Hello, everyone. My name is Sajin Zhang, and I am a master's student in the Department of Environmental Energy Engineering. Today, I will present a paper titled Electricity Generation in an Air Cathode Single-Chamber Microbial Fuel Cell with and without a proton exchange membrane. This presentation will follow the structure below. First, I will introduce the overview of the paper. Then, I will discuss the results in detail. Finally, I will summarize the key findings of the study. Let's now look at the main results of the study. This table compares power density and coulombic efficiency for glucose and wastewater as substrates, with and without a proton exchange membrane, PEM. When glucose was used as a substrate, power density was 262 milliwatts per square meter with the PEM and increased to 494 milliwatts per square meter without it. Coulombic efficiency decreased from 40 to 55% with the PEM to 9 to 12% without it. When wastewater was used as a substrate, power density was 28 milliwatts per square meter with the PEM and increased to 146 milliwatts per square meter without it. Coulombic efficiency was 28% with the PEM and 20% without it. These results clearly demonstrate that removing the PEM significantly boosts power generation, especially when glucose is used as a substrate. This paper has two notable features. 1. It evaluates both configurations, with and without a PEM. This is a unique approach, as previous studies largely focused on systems with a PEM. The study explores the feasibility of PEM-free MFCs, an area not widely investigated. 2. Performance improvements without the PEM highlight cost reduction potential. Removing the PEM significantly reduces system costs, making MFCs more economically viable. PEMs play several critical roles in MFC systems. 1. Selective proton transfer. They maintain electrical neutrality by allowing only protons, H, to pass. 2. Preventing oxygen diffusion. By blocking oxygen diffusion from the cathode to the anode, PEMs help maintain an optimal anaerobic environment for microbial activity. 3. Physical separation. They separate the anode and cathode to prevent substrate and ion mixing, ensuring system stability. However, removing the PEM introduces potential challenges. 1. Disruption of current generation. Proton transfer becomes less efficient, which can interrupt electron flow. 2. Oxygen diffusion. Oxygen entering the anode chamber can reduce microbial activity and substrate efficiency. 3. Substrate and ion mixing. The absence of a physical barrier can lead to system inefficiencies. This concludes the initial section of my presentation. I will now move on to the detailed discussion of the results. Schematic of MFC. MFC structure. Chamber. Cylindrical plexiglass, 4 cm long, 3 cm diameter, 28 ml volume. Electrodes. Anode Tory carbon paper. Cathode PEM, EFET 117, on flexible carbon fiber. Electrode prep. Cathode coated with 0.5 mg per square centimeter point catalyst, PEM attached via hot pressing. Alternative cathode. Without PEM, uses rigid carbon paper, point loading, 0.35 mg per square centimeter. Experimental setup. Microbial source, domestic wastewater bacteria. Substrates, wastewater or glucose, 170 to 200 mg per liter. Conditions, constant temperature 30 degrees Celsius. To analyze performance, we measured glucose concentrations in COD and conducted precise analyses using gas chromatography, GC. Additionally, microbial observations were performed using scanning electron microscopy, SEM. For more detailed information, please refer to the paper. Now, let's examine the experimental results and their implications. Graph A illustrates the voltage changes in the MFC with a PEM. Wastewater was replaced twice, and the system stabilized at 0.14 V for approximately 10 hours. Graph B shows the results for the MFC without a PEM. Wastewater was replaced 4 times, and while the stabilization time was longer, the final power output was 5.2 times higher than with the PEM. Why was electricity generation from wastewater so high in this study? 1. Improved oxygen diffusion and cathode performance. With a PEM, oxygen diffusion is limited. Without the PEM, oxygen freely diffuses to the cathode, enhancing the oxygen reduction reaction. Oxygen acts as a critical electron acceptor at the cathode, accelerating electrochemical reactions. 2. Reduced internal resistance. The PEM introduces additional resistance due to its material properties. Removing the PEM decreases internal resistance, facilitating smoother proton and electron transfer. 3. Characteristics of mixed substrates. Wastewater contains a variety of organic compounds and microorganisms, creating an environment ideal for microbial metabolism and electron production. 4. Use of air cathode technology. Air cathodes utilize ambient air as the oxygen source without the need for additional air supply systems. This approach increases oxygen availability and significantly boosts power generation. When glucose was used, the MFC without a PEM achieved a maximum voltage of 0.52 V, which was higher than with the PEM. However, the voltage dropped sharply after reaching its peak, indicating a decline in efficiency over time. Why was electricity generation from glucose so high? 1. High-efficiency substrate properties of glucose. Glucose has a simple chemical structure and high energy density, making it easily metabolized by microorganisms. This process releases a large number of electrons, maximizing electricity generation. 2. Advantages of PEM removal. Removing the PEM reduces internal resistance and increases oxygen diffusion, which further enhances power density through rapid glucose metabolism. 3. Efficient electron transfer. Glucose metabolism follows a straightforward pathway, minimizing electron loss. This improves electron transfer efficiency, resulting in higher Coulombic efficiency. 4. Enhanced oxygen reduction reaction at the cathode. Glucose generates stable electrons with minimal byproducts, strengthening the oxygen reduction reaction at the cathode. 5. Effect of glucose concentration. As glucose concentration increases, electricity generation also rises. Without the PEM, the system reaches saturation more effectively. The maximum voltage in the PEM-free MFC was 0.40 V, with a half saturation constant of 103, higher than that of the system with a PEM. Let's look at the impact of oxygen transfer on system performance. Results analysis. Oxygen diffusion was 0.05 mg per hour with the PEM but increased to 0.187 mg per hour, about 3.7 times higher, without it. This improvement enhanced the oxygen reduction reaction, leading to significantly higher power densities when the PEM was removed. Reasons. 1. Role of the PEM in restricting oxygen diffusion. The PEM facilitates proton transfer but physically blocks oxygen diffusion. Without the PEM, oxygen can freely reach the cathode, promoting reduction reactions. 2. Activation of the oxygen reduction reaction at the cathode. Oxygen acts as the primary electron acceptor at the cathode, enhancing electron flow and power generation. 3. Oxygen diffusion into the anode and substrate loss. Without the PEM, some oxygen diffuses into the anode chamber, leading to non-electric substrate consumption. 4. Biological factors. Biofilms on the cathode regulate oxygen consumption and support the reduction reaction. The maximum power density of the MFC without a PEM was 494 milliwatts per square meter, at 465 ohm, compared to 262 milliwatts per square meter, at 218 ohm, with the PEM. Removing the PEM significantly improved oxygen transfer, enhancing cathode performance and increasing power density. However, the diffusion of oxygen into the anode led to substrate loss, which may pose challenges for long-term operation. The open circuit potential, OCP, of the anode was approximately -0.469 V, remaining nearly identical regardless of the presence of the PEM. In contrast, the cathode OCP showed a difference. It was -0.031 V with the PEM and 0.230 V without it, a gap of 0.177 V. Let's analyze how electrode potential influenced system performance. Results analysis. The cathode OCP was 0.226 V, NHE, with the PEM and 0.425 V without it, showing a significant increase. The anode potential remained stable regardless of the presence of the PEM. Reasons. 1. Effect of the PEM on cathode potential. While the PEM facilitates proton transfer, it also increases internal resistance, limiting the cathode's electrochemical reactions. Removing the PEM reduced internal resistance, leading to a rise in cathode potential and increased power generation. 2. Enhanced cathode performance. Without the PEM, oxygen freely reaches the cathode surface, greatly enhancing the oxygen reduction reaction. 3. Stability of anode potential. The anode potential is determined primarily by the electron transfer capability of the biofilm, which remained consistent regardless of the PEM. 4. Effect of reduced internal resistance. The removal of the PEM facilitated smoother electron flow, improving cathode potential and overall system efficiency. Using SEM images, we observed biofilm formation on both the anode and cathode. The biofilm on the anode showed a uniform structure, while the cathode biofilm exhibited a heterogeneous structure. Let's examine how biofilms influence power generation. Results analysis. The biofilm on the anode facilitates substrate decomposition and electron transfer to the electrode. The biofilm on the cathode consumes oxygen and supports the oxygen reduction reaction. Reasons. 1. Role of the anode biofilm. A uniform biofilm structure enhances electron transfer efficiency and maximizes the contact area with the electrode. 2. Characteristics of the cathode biofilm. The cathode biofilm supports oxygen reduction by consuming oxygen but can lower efficiency if oxygen is excessively consumed. 3. Effect of the PEM on biofilms. Without the PEM, oxygen diffusion into the anode affects the biofilm's structure and function. Conclusion. Biofilms are critical in facilitating electrochemical reactions and determining power generation efficiency. Anode biofilm supports efficient electron transfer with its uniform structure. Cathode biofilm requires optimal oxygen regulation to balance oxygen consumption and reaction efficiency. In conclusion, this study highlights the following key results. 1. The air cathode MFC without a PEM achieved a power density of 146 milliwatts per square meter using wastewater as a substrate, significantly higher than previously reported values. 2. When glucose was used as a substrate, the MFC without a PEM recorded a maximum power density of 494 milliwatts per square meter, outperforming the system with a PEM. 3. Oxygen diffusion increased by a factor of 2.7 without the PEM, contributing to higher power production. 4. While PEM removal significantly enhanced power generation, the issue of substrate loss due to oxygen leakage requires further research and optimization.